Welcome, everyone. We're delighted to have you join us for today's webinar with REGENXBIO, who will be sharing the latest results from their AFFINITY Duchenne pivotal study with REGENX-202 in patients with Duchenne muscular dystrophy. I'm Michael Kelly. I'm the Chief Scientific Officer at Cure Duchenne, and I'm going to get us started with a bit of housekeeping. First, you'll have an opportunity to ask questions today, and you can click on the Q&A button at the bottom of the screen, submit a question anytime throughout today's presentation. We will have a moderated Q&A session at the end of the webinar, and I'll be joined by my colleague, Dr. Liana Lando, Cure Duchenne's Vice President of Research. For those questions that we're unable to get to, we'll work with the REGENX team to follow up with you after this webinar. And finally, after today's webinar, this will be available on Cure Duchenne's website as a resource for the community in case you'd like to share it with others. So let me begin. REGENXBIO's approach to gene therapy as a treatment for Duchenne utilizes a proactive, short-course immunomodulation regimen designed to improve safety, along with a unique microdystrophin gene that has been specifically designed to include an extra feature of the important C-terminal region of the gene. Consequently, the RGX-202 microdystrophin is larger than other microgene constructs, and it's not surprising, therefore, that the clinical trial has been closely followed to understand how the enlarged microdystrophin gene and the immune modulation protocol perform in patients. The company is currently recruiting the pivotal phase 3 portion of the trial and is expected to complete the enrollment by October of this year, following which they plan to file for a biological licensing application in the United States using the accelerated pathway in the middle of next year. To talk about this and much more and the recent results, we have Dr. Naz Dastgir, the clinical development lead at REGENXBIO, to update us on the latest progress from the AFFINITY Duchenne trial. So without further ado, I'll hand it over to Naz to get us started.
Hi, and thank you so much, Dr. Kelly. It's a real pleasure to be back again. I hope that my screen is showing up okay for all of you. And we're going to go through our study, kind of give you some updates that I hope you'll appreciate. And obviously, any questions you have, we're happy to take and address at the end of the call. So today I'll be talking about the AFFINITY Duchenne study, which is a trial of the investigational RGX-202 gene therapy for Duchenne. These are our forward-looking statements. RGX-202 was designed to, and I apologize, sometimes this thing shows up here on the top of the screen. There we go. RGX-202 is an investigational AAV gene therapy designed to address the lack of functional dystrophin. So we use an AAV8 vector, which is designed to deliver the transgene to skeletal and heart muscles in the body. A promoter known as SPC512, which is intended to drive the expression of the transgene in muscles. A microdystrophin transgene, which is unique to REGENXBIO. It's expected to encode a novel protein designed to retain key elements of the full-length dystrophin protein, including the C-terminal domain. So I know you all know this, but just to reiterate, the dystrophin protein is too big to package into these vectors. So you have to have a tailor-made transgene with unique portions that you think would be essential to maintaining its structure once you give it via the vector therapy. And RGX-202 is the only microdystrophin gene therapy with the functional elements of the C-terminal domain. So we use AAV8, and our promoter, SPC512, is a bit smaller, so it allows us to package a bit more in. And so how we uniquely tailored our microdystrophin is to add this C-terminal domain, which binds to the dystrophin-associated protein complex that lives in the sarcolemma or muscle membrane. And this is just a schematic here that shows RGX-202 compared to the other products that are out there, either commercially available or on a research basis. Preclinical studies indicated to us that the C-terminal domain in the RGX-202 microdystrophin preserves muscle health and enables muscle resilience. So these were studies that we did in mice. And we wanted to test how our transgene did with or without the C-terminal domain, and whether or not it was worth incorporating that portion into the transgene. How we thought the C-terminal domain might contribute to improved outcomes included prolonged microdystrophin activity. So the C-terminal domain significantly enhanced restoration of the dystrophin-associated protein complex, stabilizing the microdystrophin in muscle cells, and potentially allowing it to accumulate at higher levels and act for an extended periods of time. And it also helps with muscle health in that the C-terminal domain protects better against contraction-induced damage, enabling better muscle recovery. So these were all studies that we did in mice. And the figure here on the right shows that a few different waveforms here. So if you look, the y-axis, or this axis going up and down, is the percent initial force. And on the x-axis, we have eccentric contractions. And eccentric contractions are known to trigger muscle breakdown. So we would give the mouse about 20 contractions and then see what percentage of their initial force they were able to generate after the 20 contractions. In the mice who did not have any Duchenne, these are listed as the control mice here with the yellow, white circles. You can see that after 20 contractions, the force that they were able to generate was around 70%. In the Duchenne mice, it dropped down to below 20%. In the Duchenne mice who received our microdystrophin without the C-terminal domain, it went down to around 35%. But when we added the C-terminal domain, it went up closer to 50%, 55%. So definitely early signs that this is a good thing to add. And so that's why we designed our product the way we did. So after doing the preclinical studies on the mice, we proceeded to move on to our initial phase 1/2 safety study, and I'm able to present data on the phase 1/2 safety study for you today. So when we were addressing the safety of our product, we took a few things into consideration for our therapeutic approach as we moved into human studies, which is we obviously have the product that we talked about using AAV8, our muscle-specific promoter, which is the SPC512, and the C-terminal domain, which we preclinically felt that actually amplified the benefit of the transgene. And then we said, okay, and this was not based on any preclinical studies because our preclinical models didn't even have any immune suppression. This is based on us learning from prior people who went ahead of us and designing an immune suppression regimen that was proactive and meant to be prophylactic against any severe adverse events. We opted to kind of approach this head on so that we wouldn't have to deal with trying to play catch up later with the adverse events that could potentially happen. So we developed a proactive immune suppression regimen and implemented this from the outset of the program, and it was designed to improve safety outcomes in our study. And thanks to the participants who were enrolled in our phase 1/2 portion of the study, we tweaked the timing of our dosing. We adjusted how we were going to give this. We watched how our patients responded to this, and we were comfortable with how they performed, how they tolerated the immune suppression, and obviously our safety profile. Additionally, we have a lot of pride in the fact that our manufacturing is great. We do our own manufacturing, and we lead in purity levels in our gene therapies. So when you create your gene therapy, you are looking for a vector that's a capsid that includes the transgene product. But when you create this drug, sometimes there can be empty capsids or partial capsids, and that gets calculated into how much load you're able to give to the patients. And we have a great amount of, we have fewer empty capsids and thus a lower vector load, kind of giving you more bang for your buck in terms of how much drug you're getting per vector load. Now, today, this is a new slide. Those of you who've attended webinars before may have not seen this, so I hope this is educational in a lot of ways. So the REGENXBIO immune suppression regimen is designed to mitigate adverse events observed in other gene therapy trials. So again, like I said, this was not something that we noted preclinically, or like this was implemented even before we started dosing with our first patients. This is something that we put into place and thought that this would be something that prevented safety events down the line. And this was because we knew the trajectory of typical immune responses once you give a viral load as part of vector gene therapy, AAV gene therapy. So if you look at this slide here, this is taken out from a great paper by Laurent Servais with the ENMC. The references are listed below. Obviously, if you need copies of these papers, we're happy to share with you. But we kind of tweaked it to make it a bit more easy to understand. So you give gene therapy, and basically within hours of receiving gene therapy, you're going to start to have part of your immune system activated. And that part of your immune system is known as complement, the complement system. And that complement system is not very specific or targeted, but it's there to kind of fight anything that's foreign. And honestly, if your complement doesn't get activated, there's something wrong with your immune system. So your complement system gets activated within hours and it's triggered. And that complement, in response, even though it's doing a great job from an immune system standpoint, could have some downstream effects that are negative, which could include thrombocytopenia, which is low platelet counts, or thrombotic microangiopathy, which is one of the disorders that you see sometimes associated with kidney failure or liver injury in kids who receive gene therapy. Now, once the complement system kind of wanes off as that initial response, then, even at the same time while it's still elevated, you get this adapted immunity response, which is where you get your T cells and your B cells being formed. And this is all part of your immune system. The B cells can form antibodies, which again would be great and would be totally abnormal if you did not form these. But in the context of receiving a gene therapy, sometimes things can be a little detrimental. So what we knew is that around, for example, day to day 10 to even around month 4, in response to this boost of B cells and T cells, you could potentially have what they call anti-transgene mediated events. So you start developing antibodies to the transgene that's delivered to your body. And with that, you could still get liver injury, and you can also get myocarditis or myositis. And myocarditis refers to an inflammation of the heart, and myositis is inflammation of the muscle. So we knew this. We learned about this. And we said, okay, so what can we do to proactively prevent that from happening? So we decided to use 2 additional drugs to what's been conventionally utilized in gene therapy. That includes eculizumab and sirolimus. So every other gene therapy that's out there pretty much uses additional steroids that usually start on day -1 and are of various doses. Our dose is 1 milligram per kilogram per day. And we start that on day -1, which is the day before you receive gene therapy. But to address the complement-mediated events, we initiate eculizumab. Eculizumab is a reversible complement 5 inhibitor. And we give that to kind of suppress the amount of complement that is made in response to the amount of vector being given. Then we start on sirolimus. So the timing of the eculizumab and the dose of the eculizumab depends on the child's weight. But on average, we start that around day -9. And then we start sirolimus on day -7. And we get to a target level that we think is enough to suppress your B and T cell response. So we have that on board. That's going to also help with that second curve that you see, that second waveform. And then day -1, we start the conventional additional steroids. So this is how we came up with this regimen, and this is what we're trying to prevent. We have another slide here that's a little bit of a schematic to kind of make it a bit more patient-friendly to explain how the immune suppression works. So kind of hitting home what I just talked about before, when you give your viral gene therapy, an immune response is triggered and your complement system gets activated. And that's like a non-specific, we're going crazy, like fighting off on this vector that's coming through. And as I said before, totally normal to have a complement response, okay? That's your body's defense mechanism. It's completely natural and appropriate. But sometimes it goes out of hand, especially when you're getting a high vector load. The other thing that gets triggered is mTOR, which is the mammalian target of rapamycin. And this is something that's like a go signal. And it says B cells and T cells, go. Okay. So what do your B cells do? Your B cells are the ones that make antibodies to the transgene, and the transgene being our RGX-202 transgene, and the vector, which is the AAV8 vector. And with that, which is in that second waveform that I showed you earlier, you get the risk of myositis, myocarditis, or liver injury or failure. And as I mentioned before, with the complement system, prolonged complement activation comes with risk, especially if it's really high. And that includes the thrombotic microangiopathy, the low platelets, liver injury or failure, and sometimes acute cardiac injury. So we give the eculizumab to prevent too high of a complement response. We give the sirolimus to prevent the mTOR from being given the go. And then you doubly attack that with the prednisone that's on board. And you do your best to try to prevent the safety signals that can come about from this natural response to the vector. And we've been very pleased with how our patients have performed in the setting of this immune suppression. So here's our phase 1/2 interim safety data. So this is our portion of the study that was aiming to get early safety data and primarily safety data and early efficacy data for our patients. And we dosed a wide variety of ages and weights, and we had no serious adverse events listed as SAEs. AESI means adverse event of special interest, which we had earlier described as central or peripheral neurotoxicity, which is like a toxic injury to the nerves or brain, drug-induced liver injury, thrombocytopenia or low platelets, and we have since added myocarditis and myositis to our AESI regimen as well. And the most common drug-related adverse event that was reported was nausea, vomiting, and fatigue. So in all, RGX-202 has been well tolerated in phase 1/2 patients at both dose levels because we did do some dose evaluation, dose level 1 being our lowest effective dose and dose level 2 being our now selected dose. And we had no SAEs or AESIs. We wanted to emphasize on today's call that we are open and enrolling. Where we've been. So the phase 1/2 portion of the Affinity Duchenne study evaluated RGX-202 at 2 dose levels, and we have a positive interim safety profile that was observed. Where we are today, we are actively enrolling in the phase 3 portion of the study. So we've passed that hump of getting over like, is our drug safe in humans? Thank you so much, as we mentioned here in the lower bar, with gratitude and support to the families that took that initial leap with us to participate in that portion of our study. So we were able to get over that hump and now get into the phase 3 portion of our study where we're primarily, obviously still concerned about safety and monitoring that closely, but primarily focused on efficacy. We expect to enroll 30 patients for our accelerated approval portion of our study, but we will continue to plan to enroll beyond that because we want to complete enrollment for our confirmatory study, which is approximately 60 patients. The phase 3 portion of our study will evaluate RGX-202's impact on biomarkers of transgene expression, such as microdystrophin levels, and motor function outcome measures over time. And what's next, with the help of any patient who decides to continue to participate in our study, and the data that we get from your participation in that study is that we expect to submit a biological license application or BLA under the FDA's accelerated approval pathway in mid-2026. So I'll tell you a little bit more about this phase 3 study and how you could be enrolled or what the eligibility criteria are. So this is for boys who are 1 and older at screening. We need a genetic confirmation of the diagnosis of Duchenne. We take all mutations but those patients with deletions or point mutations in exons 8, 9, or 10. We hate excluding patients from our studies. This is because this is a higher risk region. That being said, we have expanded to include more than we initially started off with, and our intent is to try our best to establish safety even further with some of the higher risk mutations that we now allow for enrollment, and then feel comfortable to dose kids with 8, 9, and 10 deletions and point mutations in the future. But at this time, unfortunately, those are the mutations that we're excluding, but it's not like we've forgotten about those patients or don't intend to dose those in the future. We do hope with all of our hearts that we can do that. We also ask that the patients have no pre-existing antibodies to the gene therapy. And the key eligibility criteria for those who are 1 to less than 4 years of age is that they be able to walk 10 meters without assistance, have a weight greater than 10 kilograms, and they may be stable on a stable dose on or off corticosteroids for 12 weeks prior to dosing. So if you have a 1-year-old child who's not yet on steroids, because that's not what's usually recommended at that age, and you want to enroll in our study, completely fine. We just ask that you try to stay off steroids for the 2-year duration of our study so that we can compare your baseline off steroids to your follow-up off steroids. And if you decide to be on steroids, we then also ask that you remain on steroids. If your baseline testing was done on steroids, that your follow-up testing gets done on steroids as well. So that the one factor that's different between your baseline and your follow-up exams is RGX-202. The reason why we have a weight limit here of having a minimum weight of 10 kilograms is that we do safety monitoring during the study, and there is a limit to how much blood can be taken for a clinical research trial. So we have to limit the weight to 10 kilograms at a minimum. For those kids who are 4 and older, they need to be able to walk 100 meters without assistance and be on a stable dose of corticosteroids for 12 weeks prior to entering into our study, and have an NSAA greater than or equal to 16 at baseline. Like we mentioned before, we have a proactive immune modulation regimen. And our pivotal trial endpoints, the primary endpoint of our phase 3 study is the proportion of patients with greater than or equal to 10% of RGX-202 microdystrophin levels. Our secondary endpoints include time function tests, such as the velocity of time to stand, which is getting up off the floor on your back and then standing and measuring that time period. The North Star Ambulatory Assessment for 4 and older patients, the Peabody Developmental Motor Scale, and the Stride Velocity 95th centile for those kids who are 1 to less than 4. And obviously safety is still a concern for us. And our exploratory endpoints for the 4 and over patients include the SV95C and skeletal and cardiac MRIs. So we have been thankful for our team at REGENX. I have to say we have an operations team that's working with a heart of gold here to get as many sites up and running as possible to make it easier for you to access locations closer to you to enroll in our study and have more ability to get into a study as quickly as possible. So compared to our prior webinars, you might see that the list of sites has expanded. Briefly, I'll just say the names of the different sites. The physicians are here. They should all be on clinicaltrials.gov or listed soon once they become formally, once we have formally updated the clinicaltrials.gov. But the sites are Oregon Health and Sciences University, University of Iowa Healthcare Medical System, Lurie Children's in Chicago, UMass Chan in Worcester, Massachusetts, Virginia Commonwealth University in Richmond, Virginia, Children's Hospital of King's Daughters, Norfolk, Virginia, Rare Disease Research at Atlanta, Georgia, Arkansas Children's Hospital, University of Texas Southwestern Medical Center, Children's Hospital, Colorado, and Stanford School of Medicine. And we're also so happy to also state that we have activated a site in Canada, and we have plans to activate 4 additional ones. So hopefully you'll see more of Canada on the next map. And the site that we have in Canada is Children's Hospital London Health Science Center. So if you're interested in our study and you contact a site and they said, okay, you need some antibody testing, you could get it as part of the study itself, or you can go prior to even getting to the site to see if you have antibodies to AAV8 by utilizing our Affinity Beyond study. So, what's currently in clinicaltrials.gov does not reflect the most recent update that happened in the last 2 weeks, which is our key eligibility criteria for enrollment in the Affinity Beyond study is 0 to 25. So you're good to go. If you're older, that was not the case. Since we expanded to do dose, we expanded the ages of patients that were dosing in our study. We expanded the ages that were testing antibodies as well. So this is a non-interventional study seeking to understand the prevalence of AAV8 antibodies in boys with Duchenne and identify those who may be eligible for Affinity Duchenne and other future trials if you're interested. This involves a central PI who is located, or central primary investigator, Dr. Fawn, who is located in Atlanta, and she or one of her staff will contact you for a phone or video interview. And then we set up a home health visit where a nurse will come to your house and blood work will be performed and then sent off for testing for antibodies. And then Dr. Fan will get those results and she'll share them with you and the referring physician who sent you to the Affinity Beyond study, or just to you or anybody that you may ask to share the results with from that study. The key eligibility criteria here, it says 0 to 12, which is the official on clinicaltrials.gov, but it is 0 plus. We're active for that. So that will not be a barrier. And the child must have a diagnosis of Duchenne and not have received any prior gene therapy product. So in summary, I just want to emphasize that the phase 3 portion of our study is, past the safety establishment phase, focused on efficacy, but still monitoring for safety regularly, is an open label trial. So again, emphasizing no child on this study will receive placebo drug. The children in this study will all receive drug. The results of how your child performs will be compared to an external natural history control cohort. So again, we're focused on efficacy, we've established safety, and there's no placebo. We are open and enrolling. So plenty of space for you to come in. Like I said, we're going straight through accelerated approval to confirmatory enrollment for approximately 60 patients. Do not feel like we are packed. Please reach out to these sites. Please, if you have an interest, reach out to these sites, or you can reach out to us at Duchenne@REGENXBIO.com. You could look at our website at REGENXBIODMDtrials.com. And if you want to speak with a patient navigator who can also help you verify whether or not your child may be eligible for the trial, there's another website that you can access, which is a company we work with called My Tomorrows. It's https://mytomorrows.com/trials/affinity-duchenne/en-us. So we put that in place to try to make things easier for you. So please feel free to utilize that resource. And we're excited for any of you who might be interested in enrolling in our study. So I just want to take the time to say thank you so much. Mentions those same references here. There's also QR codes that you can look at about our different studies, Affinity Duchenne and Affinity Beyond. And you can also look at the clinicaltrials.gov trial listings as well. So thank you.
Naz, thank you. That was a great walk through the presentation. I particularly enjoyed the immune suppression slide, and I particularly enjoyed you taking the time to walk the community through that. They're really trying to understand both your thinking as well as sharing the data with what you've seen so far in safety. I know Leanna's just joined. Welcome, Leanna, to help us through some
Hello,
questions.
everyone.
But if I may take the first question, do you think that there's a contribution from this immunosuppression regimen into the benefits that you're seeing, the increased amount of microgene that's in there? Is there a contribution in there from early immune suppression?
So this is just speculation for me at this point, but definitely something we're going to look at in terms of, well, it would be hard to know for sure because it's speculation because we're not giving our drug without immune suppression to some patients and with immune suppression to some patients. So to definitely rigorously academically test that would be difficult. But that being said, if you are delaying the antibody response to the vector, it makes logical sense that your vector has more time to develop the transgene, and then hence you get more protein expression.
Yeah,
So
I've often tried to.
theoretically, it helps with transduction,
I
but
tried to work my way through this. The data that you've previously reported for efficacy is really exciting, and I don't know what contribution is that from the extra C terminal and the enlarged microgene, or if there is a contribution from the immune suppression, allowing the virus to actually do its work better. So it's, for me, I know I can't tease it out, but it's always a question that's going to be in there.
yeah. Yeah. So yeah, we would have to do like a separate study to test it,
Hmm.
separately. But I mean, preclinically, we see that the C terminal provides benefit, and that's in the absence of immune suppression. So you would hedge a bet that, with immune suppression on top of that, it would work even better. But
Yeah, I'm not a betting guy, but I put a few dollars on it. Leanna, you've got some questions there?
Yeah, sticking on this topic, since some things are coming in, I think people are really interested in the immune suppression, especially in wake of recent safety events and it's sort of in everybody's awareness. So there's been a couple questions about whether or not you're testing how people respond to the immunosuppressant, like how effective it is in them, if there's studies that you do before you do the dosing. And there's a question here, has been there any occurrence of an SAE from the immunosuppressants themselves?
yeah, so testing the efficacy of the immune suppression. So I can say that we do measure a very specific complement in our study called C5b9. And you can see the benefit of the eculizumab on the C5b9 for sure. For the sirolimus, I would say that probably requires some additional blood work that we don't have the capacity to draw because of blood volume restrictions. So we can't really see the benefit of that. Um, you might see some other studies state that their complement levels, went down. That's because they might be testing other complement levels like C3, C4. So it's a little different to kind of compare between different companies and complement levels, but C5b9 is a way to check the efficacy of the eculizumab. Um, the other thing is that there's something called CH50, but we don't measure that in our CIS study, but that should be decreased in the setting of eculizumab as well. But that's just extra blood work that we don't have room for. In terms of side effects from the additional immune suppression, the additional immune suppression has been really well tolerated, I can say. We don't have a rigorous data cut to go through the AEs and like related versus not. I'm sure we'll be able to report that in the future. I can say that it's been really well tolerated. Some kids don't like the taste of the sirolimus, so we go to a pill instead. I think that's the biggest issue. Chocolate syrup helps too.
Whatever makes it work.
Yeah.
Do you have any other additional questions on this topic, Mike, or should we move on?
No, please go ahead. I'm good.
Okay. So I think, we've got a real big range of questions here. So I'm
Yeah.
going to start at the beginning and
Yeah.
say, so there's a lot of questions of like, who can get this therapy? So first off, can you get this gene therapy if you had previous gene therapy? And the answer to that obviously is
Yeah.
no.
Yeah.
Yeah,
Unfortunately, it's no. Yeah. For now. I mean, everybody's
exactly.
working on ways to try to do that. And it's really for your safety and also like, risk benefit ratio. First of all, if you have antibodies from a prior gene therapy, there's a risk that the antibodies will go even more haywire if you got a second dose of gene therapy. And then that would be in the setting of your antibodies destroying the gene therapy as it's coming in. So there's no real benefit to redosing if you have high antibodies. Now, if there's a way to get rid of those antibodies, that's a different story. So
Sure. And when we're talking about antibody levels, we can kind of sort of, let's get into that topic and split it up in terms of if you've had gene therapy, now your antibody levels are orders of magnitude higher than if you've had exposure just in the environment to.
yes. Oh,
19.
yes.
Yeah,
Yes.
so maybe since we're on this topic, you can talk a little bit about, what you guys are thinking about for individuals who are above the cutoff of having pre-existing antibodies. And, some of this I recognize will be forward thinking, but, what are your thoughts around that?
Yeah, our cutoff currently is really low. You're really either positive or negative. It's like a less than 1 to 20 at this point. Um, we are actively looking to see if we could increase that in a safe way. Uh, so we're looking at some data right now, but also considering additional trials within our company to see if there's ways that we can increase the titer count, which would again be safe, and also allow for good expression of the drug, because you don't want to introduce the risk of gene therapy without even being able to deliver any microdystrophin, because the antibodies would be destroying the drug as it gets infused.
Mhm. And if you don't know whether or not you have pre-existing antibodies, there's kind of two pathways that you could go to get answers, right? You can try enrolling in the trial and be tested through that, or
Mm-hmm.
you have this study, Affinity Beyond. Maybe you could speak a little bit, because there's some questions about if you go to Affinity Beyond and seem to be clear in terms of antibodies, can you then easily switch over to the trial, or are these two
Yeah.
separate things? Maybe you could talk about
Yeah,
if,
totally.
how
So,
you might orient towards which path to take.
yeah, no, great question. Okay, so if you go to Affinity Beyond and your antibody testing is done within the 60 days prior to dosing with the gene therapy, you're good to not have to repeat it. But if it's beyond that, which can happen because, sites take a while to get to know patients and get them enrolled and whatnot. If you go beyond that, you would just have to be retested and make sure you didn't what we call seroconvert to becoming positive by the time you came into that site. The other option that we just put through with another amendment is a pre-screening that you could do at a site, where you would, like, if you're part of a clinic that's now part of a trial and you're interested in receiving this drug, you could get pre-screened at the site for the antibodies, and you don't have to go and contact Affinity Beyond to get that all set up and whatnot. So that might streamline the dosing faster for the patients in that way, theoretically, if that site does not have a wait list for patients to get into the study.
Great. Um, should I keep going, Mike, or do you want to
Yeah,
hop
no,
in?
I'd like to add a comment actually onto this part of the
Yeah.
discussion because it's really just for the community. Uh, Environmental exposure is something that we'll check during the screening process. And if those are unfortunate enough to be just slightly outside, there is ongoing work. Plasmapheresis is a good example of that, where it has shown to be useful to transiently reduce the antibodies to a window that might be dosed. And there's trials with that are occurring, and we'll get some data on it this year. And there was a recent report from Hansa, a European company that's got a product that's licensed in Europe called imlifidase. And they've been looking in that for individuals to reduce the levels of antibody and produce a window for dosing. And that recent report showed that those individuals, a handful of small number of individuals, I think it was three, was the recent report, they successfully reduced the antibody levels and allowed redosing with gene therapy. We're waiting for the full report on that, but there's an early report that's out. And that's just an FYI to the community that, as Naz pointed out, there is some solutions that's going to take time, and they're clearly in companies' minds right now in order to deal with that as we go forward.
Yeah, totally. Yeah.
Terrific. Thank you for that.
Yeah, sure.
Okay, so continuing on in terms of if we can talk a little bit more about what the exclusion and inclusion criteria. And I think there's a little bit of confusion around the exon inclusion. So, the first phases, it was from if you had to not have a mutation between 1 and 17. But that
Correct.
has changed now.
Yes, yes. Yeah. That's changed, and now the only exclusion we have is 8, 9, and 10 deletions and point mutations. 8, 9, and 10 duplications are allowed.
Wonderful.
Yeah.
Um, and so, anywhere else, deletions or anything else, anywhere else beyond those exons, there should not be there's not an inclusion for that per se.
Correct.
For if you have a deletion or a point mutation in 8, 9, or 10, what does it mean to say that you have a higher risk in that case? What, just for people to better understand why at the moment we're excluding mutations in those exons.
Yeah, so a lot of this work was done based on adverse events seen in prior gene therapy studies. I believe Roche did a study on the epitopes on exons 8 and 9 that predispose patients to the anti-transgene responses. It just 8 and 9 deletions in particular put patients at risk for the myositis and myocarditis. And the myositis isn't just muscle inflammation. You could theoretically even end up being weaker than how you started. So that put a lot of fear in people's minds and made everybody nervous about dosing these patients because they noticed that this was a trend among some of the patients that had these symptoms. That being said, we have a pretty comprehensive immune suppression regimen. So one would think, oh, why don't you try it with your immune suppression regimen? And I think it's a great idea. But we're very cautious as a company to make sure we take the right steps to establish safety before we go and tackle that higher risk population. But it's not like it's not in our mind at this time. The other thing about 8, 9, and 10 deletions and why duplications don't matter as much is that some folks might know that utrophin is a protein that exists in the body that is very similar to dystrophin. And some regions of the exons are more similar, have a lot of similarity to utrophin, and some do not. And 8, 9, and 10 are not very similar to utrophin. So then if you introduce your transgene, it seems more foreign to the body because of that, and they're less familiar, and then they'll see it as foreign and attack it. So again, that's another reason why we opted for 8, 9, and 10. And like I said, for now, and then we hope to hopefully address treating those patients in the future.
Thank you for that.
Sure.
I think, we all appreciate that at the moment, there's one time that you can get gene therapy until we solve that problem. And you wouldn't want to give, we're trying to narrow down who can't get it, right? But
Right.
at the moment, there's some individuals you wouldn't want their one shot to actually set them up for more risk of a safety event, or even for the therapy to be less effective in them.
Right. Right.
Absolutely. I can switch gears and talk a little bit about sites and contacting. It's wonderful that you've expanded to so many more sites, and the first one in Canada and more coming. We've got a lot of questions from individuals who live elsewhere, and they kind of fall into two buckets, and you can navigate through it. So, some is, when are you planning on opening sites? If you are in Europe, ex-US, India. And then the other is, what are the opportunities for international individuals to come to the US or Canada for those sites? And is that open? Is there any sort of travel assistance? Those kinds of questions.
Yeah, sure. So we're definitely interested in going even further. Obviously, we moved to Canada first. We are working to try to get even further outside in other countries, and Europe. There's some regulatory work that sometimes goes into starting your trial in other countries, which kind of slows things down a little bit. But we are doing our best to establish that, and all that's in the works, I can say, but we're not quite there yet. But with Canada, we'll be there very quickly. In terms of whether or not we've dosed kids that are international patients, totally have. We have. We are happy to support travel for those patients. What really plays into it is whether or not you have a visa where you can stay in the country for a certain period of time. Our biggest concern with patients from outside the US is just losing you to follow up. And it's not like we think that the parents aren't going to follow up. We think the parents will follow up 100%. But say there's some kind of visa issue that prevents you from coming back for follow up, that would be brutal. So we would want to make sure that that's all ironed out. And in our international patients that we've dosed so far, we have usually supported their stay for like about 6 months during the intense monitoring phase. And then they can travel back to their country and come back again for the additional follow-ups that kind of get tapered over time over the course of our study. And that travel is also supported as well, which includes like flights and hotels and whatnot.
Thank you. And that's
Yeah.
terrific. And actually, you touched upon some questions that have come up in terms of like what to expect for the treatment. So maybe we can just go through that a little bit in terms of like how long are people monitor, what is it like in terms of, time beforehand that you need to be sort of available and getting treatments, and then after treatment, after being dosed, the sort of the monitoring, and feel free to speak to the importance of longer term even getting all of this information.
Yeah, that's great. Okay. So the screening period involves obviously getting your antibody testing back. You have to go through your medical history and get all your baseline assessments, which include outcome measures, biopsy, MRIs if you're over 4, for example. So getting that all coordinated at the individual sites takes some time. Sometimes you could do that where you fly back and forth for the different visits and whatnot, but things happen even during that time. Like, say you're on a flight, your kid gets sick on the flight, you have a fever, you can't get your biopsy when you have a fever because they sedate for the biopsy. So that kind of might delay things a little bit, but hoping for the best, you get that, you can make maybe 1 or 2 visits during the first 60 days, or you can make 1 visit and then come back again for the study and get everything done ahead of time. We would support that travel. And then afterwards, again, remembering the waveforms I showed about the risky time period for the complement activation related adverse events and the adaptive immunity related adverse events. The complement period, we do like basically every other day visits. And then they taper to like every 4 days and then every week, every 2 weeks, maybe by like weeks 8 to 12, it's like every 2 weeks. And then after that, it's monthly. And then once you hit the 6 month mark, it's every 3 months. So that's why it's nice to try to be there for the first 6 months because and then after 12 months, it's every 6 months after that. So it's every 3 months after 6 months, and then every 6 months after 12 months. So we would work with your schedule to get all the visits aligned with the sites and whatnot, and get it all planned out. And again, like I said, the travel would be supported if you're in the trial.
Yeah. And to be clear, because this is a clinical trial, all of those tests, right, need to be
Yes,
done at the clinical trial site.
that's right. Yeah,
Yeah.
they all have to be done at the clinical trial site because we have all the labs, except for the first 14 days. Actually, those are all done locally because we want the primary investigators to have quick access to it. But other than that, all the other labs get done centrally, and they're from the same lab, so we can track it over time and make sure that they're safe.
Wonderful. Thank you. I feel like I've been monopolizing
No, not at
talking too
all.
much. Mike, if you want to jump in, I
Sure,
can
sure. I
jump
usually do this at meetings, so I know what it's like to sit quiet.
in.
So, yeah, a couple of points just to broaden out because I was going through some of the questions as Leanna was talking. The drug's likely to be approved in the middle of next year. And so, at what point now do you and the team actually think that you may broaden to non-ambulant individuals or those whose mutations are excluded right now in the current trial? Is that part of the thinking going forward?
Yeah, totally. So, I would say, once we get to an accelerated approval point and we feel stable, we will take that risk. I
Mhm.
think there's a lot of interest in doing that. So that's me pushing on the inside as quickly as possible. But logistically, I think, yeah, I think that is the logical point to take that risk at that time because we're very happy with the safety profile that we have so far, and we don't want to introduce something that's not, if there were an adverse event to happen in that population, we don't want the rest of the population to suffer because of it, in the sense that because of because of the specific mutations that are higher risk
No,
or
absolutely. Like in my words, I would say I don't want to put at risk having this drug approved and available to everybody.
right,
And at a future stage, at some point, whenever you're more comfortable, then you
right.
can start to broaden the ask various questions about specific mutations or non-ambulant individuals.
Yeah,
Yeah,
yeah.
and just to broaden on that, the 60 patient confirmatory trial, is that the exact same inclusion exclusion criteria that you're
Yes,
currently using?
yes. I
Okay.
mean, it's an arbitrary distinction between the 30 and the
Yeah,
60.
I
The
figured.
30 is the part where we're submitting for the biological license application, but we will continue to enroll, and everything is identical between the two portions of the study.
Yeah, I think that's important to get that message out. I was glad to see that on the slide today because I'd certainly been asked some questions in private about what's next. Is this going to stop at a certain point?
No,
And
no, no.
I
Yeah.
must say, and the point that Liana brought up, there has been some confusion out there about gene therapy in general. And to have that statement is an important one for the community that this is going to continue to recruit beyond the 30 in October.
Yeah, definitely.
Okay, excellent. Thank you. Liana?
We want to do we want to complete our confirmatory
Yeah,
study as quickly as possible.
totally agree.
Yeah, I think we've been covering a lot of it. I think the last sort of thing is, how for the first individuals that have been dosed, how far out are they, and what can you say about motor indications in them? I know some of this has been talked about in other webinars in public, so I feel safe asking the question here for people who maybe haven't heard it before.
Yeah, so the latest results, I don't think we've put together yet, but our first patient is now, mind you, our first three patients were dose level 1,
Hmm.
but that's the lowest effective dose. So they're still doing well. But our first patient and I think our second patient are both 2 years out, but we haven't put the data together. But they were both with dose level 1. We don't yet have 2 years of data on our dose level 2 patients, but we're getting there
Hmm.
soon. It gets a bit complicated about when we can present certain data because some of our dose level 2 patients have rolled into the phase 3 study. So we've been given the okay to present phase 1/2 data, but um, I think it's kind of messy when you're st keep statistically analyzing your patients as you're studying them. So we try not to look too much at the data as we're dosing in the phase 3 portion of the study.
Exactly right, because the phase 3 is designed to get at the motor outcomes, right, which are going to show benefit.
Right.
And if the data comes through the way, we're all hoping it does, you can then use that to go from accelerated approval to full approval.
Yes. Yes.
Exactly. So, and as you said, that's why at this beginning stage, we don't have as solid data, uh, because statistically, you don't want to compromise later data points by looking at it too much now and
That's right.
actually lowering your statistical
Statistical
power.
power. Yeah.
Yeah. And, how long will you follow individuals out?
So the phase 3 portion of the study is for 24 months. So that's when our functional endpoints are read out. So we're thinking if you even have stability, which we'll see if we have better than that, which I think we will, hopefully. This is just me guessing. But if you can prove that at 24 months, then great. So we want to give the kids enough time to kind of show that they're deviating with what's expected from natural history, because again, we don't have a placebo group. And then after that, they'll complete the phase 3 portion of the Affinity Duchenne study, but then they'll enter into a long-term follow-up study. And that long-term follow-up study is required by the FDA for anybody receiving gene therapy so that we monitor the patients for 5 years total after they're dosed with gene therapy. So they would be in that long-term follow-up study for 5 years for up to 5 years after dosing, which on average is 3 years.
Thank you. And then in terms of, there's some questions about other medications that you could take, like, people have mentioning givinostat or exon skipping drugs. Maybe you could speak to when you can do that and when you can't do that.
Yeah, so I mean, okay, so please do, I would say to any patient enrolling in the study, please avoid going on any of these medications during the first 2 years of your study. Obviously, as you know, anytime you consent to a study, you can exit that study at any point. The power is in the patient's hands. But it's to your benefit to stay in the study and be monitored and be safe in that study. So because we do our baseline assessments off all these medications and our endpoint is 24 months after dosing, we want the one changing factor to be RGX-202. If you added a different drug or took away a drug that you were on, like if you decided to go off steroids, which I know a lot of people dream about doing that, but please do not do that in the first 24 months because like if your baseline test was on steroids, we want your follow-up test to be on steroids and the only difference to be RGX-202. That kind of muddies the picture in that first 2 years. Ideally also like for the long-term follow-up, because we're monitoring safety after RGX-202, if you were to start on a separate therapy, say any of the drugs or antisense, and there was an adverse event, it'll be really hard for us to tell whether or not it was from the new drug that you added or from our drug or from a combination. So ideally, I think, you would hold off there, but I know you can't stop patients from doing what they want to do. So if they really wanted to go on to something, we would still continue to monitor them on that drug. But hopefully you wouldn't feel the need to go on to something else.
Okay, I'm going to wrap things up. Naz, I really appreciate you being here today with us and taking so many questions from both Liana and I and the
Yeah,
community.
sure.
Yeah, and it's a real big thank you to the whole team at REGENX for all the work they've done advancing this here and for sharing all the important updates. And we truly look forward to future updates as this here moves into next year with a BLA in the middle of the year through the accelerated pathway. So
Yeah.
thank you, and thank you to all of your colleagues at REGENXBIO.
Listen, I'm only here riding on like a mosh pit on the backs of all my team. I mean, there was such a heartfelt thank you from all the team members. There's a lot of work that's going. You see my face here, but there's so much behind me. And we're very dedicated to Duchenne at REGENXBIO.
Thank you. And to everybody else that's on the call, a post-webinar email will be sent tomorrow should you have any additional questions. And we'll get in touch with all attendees to let you know when the webinar will be posted and reviewed so that you can share it to those that may be interested. And with that, I'll close it down and thank everybody. Have a great rest of your day.
You too.
Bye.
Bye.
Bye all.